Hello and welcome to Nikolai's genetics lessons, and today's problem is in a cross between green leaf and purple leaf pure breed rice varieties. All the offspring in F1 generation are green, but in F2 generation progeny green and purple are obtained in a ratio 13:3. Explain. As usual, I recommend you to pause video here, try to solve this problem on your own first, and when you would be ready you can run video again and you can compare your answer with my answer and explanation. So my first hint would be that when we see 13:3 ratio, 13 plus 3 would give us 16. So this tells me that this is one of the variants of the problem with dihybrid cross with ratios that we get used to from our classical examples that Mendel did with peas in dihybrid cross, where we obtain 9:3:3:1 phenotypic ratio. But this is a modification of this ratio. So here we have 16 and here we also have 16. So this tells me that this problem involves two genes, but the ratio is unusual. And before I will proceed with explanation how to solve this problem, let me first give you an example. Those I have been unable to find pictures of the purple leafed rice. I'm going to use example of the tea plant. It is very popular plant in the place where I live in Hawaii, and this is not tea that we drink, but this is Ti. So this is Ti, tea plant. And this plant can be found in two varieties. One is green variety, another variety is, as you see, red. But what makes this green plant red? Accessory pigments. So normal, of course, color of the plant is green, and it is green because chlorophyll is green. But when there is a lot of accessory pigment, there can be different carotenoids or other accessory pigments. This plant can be found to be red or purple. So, but if we will destroy this pigment, it doesn't mean that this plant would be white. It would be green, because this red pigmentation just hides this green color, which is due to presence of the chlorophyll. And basically my whole presentation today going to be about epistasis and epistatic influence of production of this accessory pigment. We have two genes. One gene would control production of this accessory pigment, and another gene would produce this pigment. So we talking today about one gene that is suppressant and another one which produce this accessory pigment. And if no accessory pigment would be produced, the color would be green. If it would be produced, the color would be red, or as in our example, it would be purple. So let me clean the space. So basically, we start with green leaf variety. So genotype of this variety would be capital A, and small b, and dominant allele A here work epistatically and suppress dominant allele B here. But because there is no dominant allele here, so recessive allele would be non-functional. So the color of this plant would be green. So I would use green color to underline this genotype, and we cross in our first cross with purple leafed variety. So this is purple leaf, and genotype would be small a, and capital B, capital B. So we see here that gene that suppress production of the purple color is defective, so no normal allele is produced, and in this case purple accessory pigment would be produced. So phenotype would be purple. So this gene suppress production of this gene. So this is classical epistasis, and if this suppressant gene or two alleles that represent this gene would be defective, in this case, of course, this accessory pigment would be produced. So what would happen in F1 generation? So if we cross these two varieties, of course this parent only can give dominant allele A, and another parent only can give recessive allele A, and this parent can give only dominant allele B, and this parent only can give recessive allele B. So this is going to be genotype of the F1 generation, and what phenotype would be. As you see, one dominant allele here is present. That means that normal protein that would suppress production of this purple color would be produced, and this genotype should produce purple accessory pigment. This suppressive allele would cancel function of this dominant allele, and the color would be green. So 100% of the F1 generation is green. So this is exactly what we see according to our problem here. 100% of the F1 generation is green. Once again, this is very important, so I will spend one more minute here. Dominant allele means that normal, maybe globular, most of the proteins are globular, would be produced, and this type of alleles would suppress production of the normal accessory pigment, which is purple. But this allele that can produce this purple color also can be dominant and recessive allele is defective. So even if we would have small a here, but these two alleles would be defective, no purple color would be produced. Purple color would be produced only if both alleles would be defective, both suppressant allele would be defective, and at least one of these alleles would be normal, that would produce normal accessory pigment. So as you see, here is exactly such genotype that would produce such phenotype. And in F1 generation, we see that one allele of the suppressant gene is normal, another is defective. We also see that for the other gene, one allele produced normal protein, which we perceive as purple accessory pigment, and another one is defective. So it doesn't produce purple pigment, but this one produce. But because here we have a suppressant gene, so we have here epistasis, when one gene control behavior or phenotype of the other locus. So basically one locus controls another locus, because this is two different genes. So this is gene is dominant over these two, and also we can say that this is epistatic and this is hypostatic. So what happens in the F2 generation? In F2 generation we cross F1 generation with itself, self-pollinate plants, so we cross with the same genotype, and of course this means the same phenotype. So this is also going to be plant that is going to be phenotypically green. And because these two parents are heterozygous, as you see, this is classical dihybrid cross with usual ratios that we see 9 to 3 to 3 to 1. But today the picture would be different. So let's find out what kind of gametes such parents can produce. And parent 1 can produce gametes capital A and capital B. This is first variant of the gamete. Second variant would be capital A and small b. So capital A and small b. And the third variant would be small a and capital B, small a and capital B. And the last variant of the gamete would be small a and small b, small a and small b. Once again, this is going to be genotype of parent 1, and parent 2 has the same genotype. That means that parent 2 also would produce the same type of the gametes. So parent 2 would produce capital A, capital B gametes, capital A and small b gametes, small a and capital B gametes, and small a, small b, small a and small b. So now when we build a Punnett square, this is going to be 4 by 4 Punnett square, 16 cells. We can find all the probable genotypes and phenotypes that such cross would produce. One more time, the first gene is epistatic to second gene, or first locus where this gene located is epistatic to the second locus. So in the first cell we have capital A, capital A, capital B, capital B, capital A here, capital B, small b here, capital A, small a here, capital B here, capital A, small a here, and capital B, small b here. Capital A here, capital B, small b, capital A, small b, capital A, small a, capital B, small b, and capital A, small a, and small b, small b. We have done capital A, small a here, capital B, capital B, capital A, small a here, and capital B, small b, small a, and capital B, small a, and capital B, small b. The last column, capital A, small a, and capital B, small b, capital A, small a, and small b, and small a, and capital B, small b, and small a, small b, small b. So we done, and once again, here we have parents, one parent here on the side, another here on the top. This is gametes, and if this is male parent, so male parent would produce pollen, and female gametes here on the side would join with male gametes, and here in the cells we would see diploid organisms. So each gene would be represented here with two alleles. So what phenotype we would see here? Phenotype of this parent would be green. Those, as you see, in the locus that specifies accessory pigment, normal to normal alleles present here, which can produce purple pigment, but here we see also normal suppressant proteins would be produced that would suppress production of this pigment, so would influence expression of this locus where these alleles on the other chromosome or on the same chromosome can be found. So phenotype of the plant would be green because initial green color of the plant wouldn't be masked by accessory pigment. And here we see the same picture, so green phenotype here. Once again, in this cell we also see that two normal alleles that can produce accessory pigment, but also we see here a suppressant allele, so this also would produce green color in plants, and this phenotype also going to be green here. So basically we are right now looking just for presence of the suppressant allele, and here we have two suppressant allele, so also phenotype would be green phenotype here, green phenotype here, and green phenotype here because dominant suppressant allele is present, green phenotype here, and here is interesting. Here, as you see, no suppressant normal protein would be produced, so this would be defective, and this plant also has two normal alleles for accessory pigment. So the phenotype of this plant would be purple, so this is going to be purple, and here we also see that two suppressant alleles are defective and one normal allele present that would produce purple accessory pigment. So phenotype also would be purple, and green phenotype here, green phenotype here because normal suppressant allele is present here, and purple phenotype here. And the last one is interesting. So what the color we can expect here? No suppressant allele, but also no alleles that would produce purple color. So we would see normal color of the plant, which is green. So phenotype here would be green. And now, as you see, out of 16 cells, 3 cells produce purple phenotype and 13 cells produce green phenotype. So we got ratio that is 13 green, 2, 3 purple. Once again, this is example where we have two genes, two loci, and one locus produce purple accessory pigment, and another locus controls, or we say epistatic, to this gene. So if this locus also would have at least one dominant allele, A, it would suppress production of this accessory pigment. If both alleles of the locus that is epistatic to another locus, so this would be hypostatic, this would be epistatic. So if both these alleles would be defective, so these alleles wouldn't be able to produce normal proteins that would suppress production of accessory pigment. And if we would see accessory pigment for the gene B, which is dominant, then we would see that phenotype would be purple. But if no suppressant protein would be produced and no purple accessory pigment would be produced, we would see normal color of the plant, so also would be green. And hence we got this ratio. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. And see you in the next video. Goodbye.